in NCBI, all the things are mentioned. All database is there. All other information are there. You have to extract or you have to retrieve the nucleotide information from that particular ID. For example, here it is there, nucleotide. I'm pasting that thing here and I'm searching this thing. So when I search this thing, I automatically get the organism, which is Gallus gallus, that is chicken, and the gene name is ATR. So here it is. So it is ATR gene, and it is present in Gallus gallus, that is nothing but the chicken. And it is which kind of gene? mRNA gene. So in case of this database, we can store our information either in the form of DNA or RNA or protein sequence. So in this case, I have retrieved the information in the form of a format that is known as FASTA format. This is there. Are you able to see this thing? Everyone? Major thing, the noticeable thing about that thing, about the database is that you cannot store anything in any format. For example, Surya has said that, sir, I have a gene, sir, that consists of this one, lots of things, this and this is this, and at last G is there. Can I store this thing? You're not. You cannot do that. For doing that thing, you need a format. And there are various kind of formats that are present in database, and the most common one are, first is FASTA format, and second one is GenBank format. The earlier name was GenBank flat file format. So in this case, lots of information are given about that particular gene or that particular nucleotide, but in this case, only DNA sequence or the sequence information is given with optional information. With optional information means, for example, it has been written here that it is a Gallus gallus ATG gene. For example, in this case, it has been written here. It is there. It is the accession number which has been given to Surya, and after that, the Gallus gallus organism is there. The name of gene is there, and the type of gene. For example, it is mRNA sequence is there. All information is given. It can be written as such, or it can be written in this form also. For example, I can change it like that. For example, greater than sign. If you want to consider ID, you can consider. Otherwise, for example, I can write it as test. After that, I can copy paste whole sequence which is there, which is also applicable in this case. So it is nothing but FASTA format, which starts with a greater than sign. The other format is known as GenBank flat file format. Here it is. So here it is. For example, I am again copy pasting that thing. I'm showing you the sequence, how you can retrieve that thing. First, you have to go to NCBI, which is ncbinlm.nis.gov. After that, you have to paste your ID, which has been allocated to you. If you are clicking on this all database thing, you will get entree search result. This entree search result will tell you 40 different categories. That how many books are there, how many mesh means medical subheadings are there, how many publication has been done regarding that, how many nucleotides are there, how many proteins are there. But as I know, because it is related to that particular gene ID, so it is giving you, it is a gene, or it has certain kind of identical proteins that are present. So I'm clicking on that. This is one way for going towards that particular gene. Or there is another way, by simply going towards, by selecting this thing, that is, I'm going again. Here it is. You have to specifically go towards the nucleotide sequence because I have to download it. So here it is nucleotide. I am pasting this sequence number, accession number again, and I am clicking on it here. So when I click on that, it automatically gives a format which is nothing but GenBank format. GenBank is a flat file. GenBank flat file format is a detailed version of that particular gene, in which it has been given what is the locus number of that, what is the accession number of that, what is the base composition, how many base pair, what is the length of that particular sequence. It is 8,419 base pair. Which kind of type is mRNA linear is there. When it is submitted, 17 May 2018, all the detailed information is there. That's why it is more, you can say, elaborative one as compared to the FASTA format. While in case of FASTA, when you click on this, it is just a brief information. What is that particular thing? Gallus gallus, ATR, or this kind of thing is there. After that, whole sequence is there. But the only recognizable thing is that in case of FASTA, it always starts with a greater than sign. Whenever a greater than sign is there, then you can simply say that till now you can say that it is a FASTA format. But there are other formats also that can also recognize as this greater than sign, but I will not tell today. But in the subsequent class, I will tell. Today, till today, you have to recognize FASTA as greater than sign. So we have to. There are two common formats that are available in bioinformatics. One is FASTA format, and another is GenBank format. So I'm going to the details of this particular thing, that is GenBank flat file format or GenBank format. As I earlier told you, in case of FASTA sequence, it always starts with a greater than sign, while in case of GenBank format, it always ends with double slashes. In this case, there is a specific format for writing this DNA sequence. In a single line, you can only write 60 base pair. So it is starting here 1 and it's ending at 60. Next one is 61 and last one is 120. Next one is 121. So in a single line, only 60 base pair can come. Also, the fragmentation is of 10 base pair only. So here 10 base pair, 10 base pair, 10 base pair. Total 6 fragments are there. So 60 is there. So in case of GenBank flat file format, this format has been followed. That is in this case, for example, 60 is there, and at last we have to classify whole base pair into this 60-60 category, and at last we have to put double slashes. Other than that, other than this identification thing, there are details are also given. For example, what is the definition of this thing, which is given in the FASTA format also. Gallus is there, gene name is there, and mRNA is there. Also, what is the accession number and what is the version number. Also, what are the keywords that are available. What are the what is the, you can say, taxonomical classification, where it belongs, eukaryotes, and it goes towards the Gallus, that is chicken. And what is the what are the number of papers that has been published regarding that. All the things, all the annotation, all the editing that has been done in this particular gene, all the things are there. What is the source? Total sequence length is 1 to 8,419. But out of that, there are certain in this particular gene, there are certain coding regions, CDS region are available, which starts from 38 and ends with 8,020. It means the whole sequence of that particular gene is not coding or not, you can say, functional, but there are certain fragment out of that which is functional, that starts from 38 to 8,020. And other than that, there are other information, for example, where it is starting. For example, it is 10, 20, 30, and 38. Here it is. The region that lies between start codon and stop codon is considered as coding region. So the whole gene is not functional, but after this thing, it is functional. It means it can encode certain kind of proteins. So we have to take two different formats. We have to first download your sequence. How can you download that? First, you go towards this nucleotide, select your ID. This ID is nothing but the accession number. I will tell you the difference between whole three things. There are three different terminologies that are commonly used in case of bioinformatics. One is known as accession number. The second is known as version number. And third one is known as GI number. This GI number has been omitted. I think last 2 years it has been omitted. First, for example, if you go towards NCBI and you want to submit your data, which data? DNA data. When you submit that data, it can be relevant or it can be irrelevant. So when you store your data, then they give you certain ID that is known as GID, GI number, that is gene ID number. It can be correct, it can be incorrect. After a certain period of time, they will email you that your gene has been selected or gene is relevant. If it is relevant, then this GI number has been converted into accession number. Are you getting this thing? What is the difference between GI number and accession number? So in this case, the accession number has been given. It means this particular gene is actually relevant in nature. So it has been allocated certain accession number. But for example, some student is working on this same gene. For example, student A has submitted this gene. He got GI number. After confirming, he got accession number. For example, he got accession number 1, 2, 3, 4 or ABC 1, 2, 3, 4. But after that, student B are also working on the same gene, and he has found actually it consists of 7 coding region which is not mentioned in that particular database. So he is actually editing that thing. When you edit certain thing which is same genome or which is same nucleotide sequence, then a version number has been allocated. For example, the earlier number was ABC 1, 2, 3. Right now it is 0.1 because one time modification has been done by student B. For example, subsequently student E has done the modification 5th time, so the version number becomes ABC 123.5. So by looking at that particular database, you can easily visualize how many number of modification has been done. How many modification has been done in this Gallus? 0.2, which represent number of modification. So by looking at the database, by looking at the GenBank you can say format, you can easily visualize what is the locus number, what is the accession number, what is the version number. Out of that, what are the organisms that are involved in it, or what is the taxonomical classification, or what is the region of CDS or coding sequence. CDS means coding sequence. whole sequence will not be coding, but there are certain fragments that is coding. So first, I will show you how to take DNA sequence in the form of FASTA format. I'm taking the example again of that particular gene. I have gone towards this. I have selected this thing. Click on send to file. Which format you want to take? FASTA format. Create a file. When you create a file, the sequence format will come. So here it is. So here I'm showing you here. So here it is. Open with. Where it is. Open with is there, and you have to click on the notepad or wordpad. You can also click on that. So here it is. Are you getting this sequence? It starts with greater than sign. It means it's a FASTA sequence. You have to download this sequence on your system. Okay. This is FASTA format. But in case of GenBank flat file format, you do not have to do like this. For example, you do not have to, here it is, GenBank format. You do not have to go towards this, either this, Control C, Control V, Control A, all the things you do not have to do. You have to just take a screenshot because you need the above information as well as below information. What is the indication of using this GenBank format? How can you identify GenBank format it is? Other than that, the sequence fragmentation is of 60 base pairs. So here it is. So this is the structure. This is the main structure, or you can say the above structure. There are two parts in case of GenBank format. This is the structure, and this is the main sequence. So first you have to take screenshot of this. For example, I'm clicking on print screen. When I click on print screen, it automatically takes the screenshot, and I'm going towards the paint. Then you have to Control V or paste this sequence. This whole screen come, and you have to selectively take the above thing. Here it is. Here it is. Cut, and after that, you have to paste it on the Word file, Word document file. So here it is. I have taken that thing. So my gene, there are two different formats today we have to deal. First is known as GenBank format, and another is known as FASTA format. This particular screenshot represents GenBank format. My question is that, are you able to identify whether it is a GenBank format or not? First, I have to take the structure part, and we have to also consider the sequence part. Go towards the end and take the screenshot of that. So here I have taken the screenshot of this, and again I have to crop this thing. So here it is. I have just taken the bottom half so that I can consider accurately. So here it is. Crop. Just enter and paste that thing. Now it is a which format? GenBank flat file format. Other than that, you either just copy paste this thing, or you have to just download this thing in case of FASTA format. So this particular sequence is very useful in your subsequent experiment when you perform all the subsequent experiment in bioinformatics. So I have taken both the format. So your first task is in today's experiment, you have to take the sequence from the NCBI. Also, you have to download a software that is known as SecuIn. So here it is, SecuIn. It's a submission tool. I have shown you how to retrieve the data from NCBI either in the form of FASTA format. Here it is, from greater than sign it is showing, and or in the form of GenBank format, which ends with double slash, or that has 60 base pair fragments in that. So I have done first thing, that is how to retrieve the data.